The actual story of how our understanding of DNA became what it is today is fairly convoluted, and it's not really the result of any one person, but decades of investigation by many different people. Arguably, the first step along this process would be in 1869 when Friedrich Miescher isolated nuclein. Uh, Basically, it was DNA with the associated proteins from cell nuclei, and he was the first to identify DNA as a distinct molecule from anything else. The next step on this path would be Thomas Hunt Morgan in 1908 when he determined that not only were genes on chromosomes and that chromosomes carry genetic information, but he's also the guy that figured out that some traits are linked to particular chromosomes. More specifically, he determined sex-linked traits by studying fruit flies. Basically, he had white-eyed and red-eyed fruit flies, and the white eyes tend to be more dominant in males, and so on, which determined sex-linked characteristics. The next would be Frederick Griffith in 1928. He was studying the pneumonia bacteria, and he discovered first that there were two major types, smooth and rough. Now, when he injected the mice with smooth and rough bacteria, he found out that the smooth was deadly and the rough was not. So what he then tried to do was he took some of the smooth and he heat killed it. And then he took that heat killed bacteria and he injected it into a new mouse. And what he determined was that the mouse survived just fine. So then he tried mixing some of the heat-killed smooth and the normal rough together and injected that into a mouse. What he determined then was that the mouse died. So this was really strange. You took something that was harmless and something that was harmless, and you mixed them together to produce something that was deadly. When he actually extracted the blood from the mouse that had died, he found that there was rough. The rough bacteria had become the smooth bacteria. So he said that, well, this means that bacteria can uptake genetic information from its environment. This process is what he called transformation and turned out to be a major step in our understanding of genetics and DNA and evolution and bacteria. So at this point, we know that transformation exists and we know that chromosomes exist. But there's still a lot of confusion because, like Miescher said, the chromosome was made of DNA and its associated proteins. And so before the 40s, most biologists thought proteins were responsible for inheritance because, hey, there's a lot of different ones that come in all sorts of different shapes, and inheritance is really complicated. How can something that only has 4 or 5 subunits really make all of the differences that we see in life? And so no one really had any idea about DNA structure or function. We knew there were different nitrogenous bases, but that was about it. And so it didn't seem possible that DNA could be the carrier of genetic information. Just 3 scientists came along in 1944 and tried to modify Griffith's experiment. They took the non-deadly rough type of bacteria and they mixed it with the heat-killed smooth. And then they put it into something where each of the vials had a very particular thing destroyed. So when they had all of their mice mixed with the various bacteria sources, they injected each of the mice with the varying combinations and determined that, well, they still died, except when the DNA was destroyed. That mouse lived. So their conclusion was that DNA is the transforming factor. They didn't take it far enough and say, okay, DNA is the carrier of genetic information. Don't think they're saying that. The only step they said was that DNA was the one thing that did the transformation that Griffith had determined. The next step came in 1947 when Erwin Chargaff determined that the bases of DNA have a very characteristic relationship. He said that, well, A binds to T and G binds to C. So they became known as what we know it now as Chargaff's rules, and it was very basic understanding. Now that actually fit with what we knew about DNA, because if you took two purines together, the structure was too wide to fit into that regular width that DNA has. The pyrimidines were too narrow. So only by mixing a purine and pyrimidine were you able to get that perfect fit. Now, he did not really understand that differences in here were also involved hydrogen bonds. That did not come along till later, but this is still an important piece of knowledge that aided our understanding of the structure of DNA. In 1952, Hershey and Chase used the T2 bacteriophage, which you can see pictured here, to try to understand it a bit more. What they know is that the bacteriophage was only consisting of two major parts, the protein coat and the DNA. So they raised this bacteriophage in an environment with P32 and S35, which are radioisotopes. DNA, if you remember, has phosphorus, and proteins does not. Sulfur is in proteins, but not in DNA. So by labeling it with this, we could tell DNA and protein apart in a way that we could not tell prior to this. So with the radio-labeled bacteriophages, what they then allowed to happen was the bacterium were placed into the same environment as the bacteriophages, and the phages were allowed to then infect the bacteria. So those infected bacteria were then spun in what was essentially a blender, and it forced the viruses to come off of the E. coli. Those samples were then centrifuged so that the mixture would form a pellet at the bottom of the test tube. So the virus protein shells and things stayed suspended in the fluid, but the heavy E. coli went to the bottom. They then measured the radioactivity in the pellet in the liquid. The conclusion here was that DNA carried genetic information. This was the first time that claim was made. So using all of this prior information, James Watson and Francis Crick came along in 1953 and built what we now know as the double helical model of DNA. Their model was, however, based on the work of Rosalind Franklin, which was given to them by her colleague Maurice Wilkins due to reasons we'll discuss in class. What she provided was known as Photo 51. And you see that X shape that's there in the picture? Well, this picture is an example of something called X-ray crystallography. And what Franklin and Watson and Crick realized eventually is that this X shape is representative of a double helix. So by using her picture, it gave them the final piece of information they need to develop their model. So now we know the structure of DNA, and we know that it is the carrier of genetic information. Just to refresh, we know that there are 4 nucleotides to DNA, A, T, G, and C. We know that these follow the rules of base pairing, A to T, G to C, as determined by Chargaff. We know that there is a sugar phosphate backbone, which allows these molecules to bond to each other. We know that the DNA is arranged in a double helix, and that this structure is DNA. And without the various pieces of information that we've developed over these many years, we couldn't know any of this. The big question now was, how does DNA copy itself? We know how it's made or why it's made this way, but how does 1 become 2, become 4, become infinity that we have today? Or what the various scientists to this point then realized is that the phosphate to sugar bond involves carbons in the 3 to 5 positions. So you end up with 5 to 3 and 3 to 5 in an anti-parallel DNA strand, where the strands are running opposite directions. And so what Watson and Crick said was that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Now, making the claim is one thing, but now you have to prove it. In 1958, Meselson and Stahl set out to do just that. They determined that the basic process of DNA replication was one of 3 possible models. It was either conservative, where the parent strands stayed completely together and made a completely new daughter strand. It was semi-conservative, where each strand split and became the template for the other. Or it was dispersive. It broke up into chunks and spread out. But we couldn't really know what was which one was it. And so they decided to take a page out of Hershey and Chase's book and use radioisotopes. They used different weights of nitrogen to determine this. The original bacteria DNA was cultured in a medium containing the heavy isotope of nitrogen, nitrogen 15. That was then transferred to a medium containing the nitrogen 14, or the normal elemental nitrogen. What they did was then centrifuge the sample after the first replication, and they determined that they had only one weight. There was a mix of 15 and 14 together into the thing. If the parent had stayed separate, I would have two bands, a heavy band and a light band, and I didn't. I had a medium band. So that knocked out the conservative model. So after the second replication, a new sample was centrifuged. And this sample, we found two bands. So that automatically knocked out the dispersive. Because in the dispersive model, I should have ended up with an even lighter band than the first time, because more of the lighter nitrogen was involved. But now I see two bands. One where the original parent, the one strand has stayed together and I have a daughter strand completing the molecule, and the other where it's made of two completely new nitrogen-based daughter strands. So this showed us that DNA replication is semi-conservative, and this is one of the major experiments in biology.